Great. Howdy, everybody. This is going to be the video for chapter 38, which is all about angiosperm reproduction, or just our plant reproduction unit. So first off, we haven't been using the word angiosperm a ton, so I first want to just talk about what is an angiosperm. So angiosperms are the flowering plants that have seeds that sort of come packaged enclosed in a fruit. And you may remember fruits are, biologically, those are the sinks, the sinks for the sugar that we learned about last chapter. So angiosperms also are going to be the vast majority of plants on land, 85% of land plants. And the big thing about angiosperms, sort of the key idea here, is that they utilize sexual reproduction to increase their genetic diversity. So first, just to break that down, how does sexual reproduction increase genetic diversity? Well, there are three things that I want you to remember about that. So the first two are going to be, you should remember them hopefully a little bit from last term, and that's going to be crossing over in meiosis. And also during meiosis, we're going to have independent assortment. And these are two principles that create a lot of genetic diversity. Crossing over, you may remember, was when we had sort of these two chromosomes, these homologous chromosomes, and they would swap segments of DNA. And then independent assortment was the property that, the arrangement of these two chromosomes lining up does not affect the arrangement of these two. And then the last one is random fertilization. So random fertilization means that the pollen, the sperm and egg that become, the sperm that fertilizes the egg is random, and the egg that gets fertilized is random, which is another way to increase genetic diversity. So next I want to get into the anatomy of the sexual systems of an angiosperm. So here we have a flower. I've sort of cut it in half so we can see the stuff in the middle, because flowers are usually sort of shaped like this. My fingers would be the petals, and then right in the middle would be the ovaries, and the anthers would be coming up out of that. So I've tried to draw that a little bit. And so these are two flowers, and I really should have drawn this as well. So this angiosperm has, first of all, it has these leaves that are right by the sort of the base of the flower. Those are called the sepals, and those are going to be important. They're going to essentially provide the energy to the sexual system through photosynthesis, right? And then we have our flower petals, which everyone knows what a flower petal is. Those are mostly going to be protective, is going to be the main function for those. And then you can see we have these little sticks with little bulbs on the end coming off sort of the middle area there. So those little sort of sticks, those sort of supportive lines are called filaments. And then the bulbs at the end are the anthers. And anthers are responsible for producing pollen. And pollen is essentially the sperm of a plant. It carries sperm cells. So that's anthers. And an anther plus its filament is referred to as a stamen. So a stamen is sort of the male reproductive organ of the flower. And then the female reproductive, it's here. Like you can see that it has both these anthers, and it has this structure. So this structure, I've drawn it over here as well, is the female reproductive organ. So it is, you have this stigma at the top that allows the pollen to bind to it and actually become saturated with water. And the pollen has these little cells called tube cells that actually create a tube that penetrates down the stigma, down the style rather. The style is sort of the shaft region. But anyway, it can grow this pollen tube that will go into the ovary. And so this is the ovary. This whole structure is the ovary, which is the female reproductive organ. and then that pollen is going to be able to deposit the sperm into the ovules of the ovary. All right, so that is just kind of the rundown of the anatomy. So now I want to get into sort of the types of flower, of the sort of sexual plant types. So first, and the one I've drawn up here, is called a perfect or complete flower, and that means that it has both stamen and carpel. And carpel is another word for this whole female reproductive system. And remember, stamen is the male reproductive organ. So anyway, perfect or complete flowers have stamen and carpels on the same flower. So in one singular flower of the plant, remember, plants can have many, many flowers. Think of like cherry blossom trees. They have tons and tons of flowers. So a perfect flower has both stamen here, the male reproductive organs, and it has your carpel, or your ovaries. So imperfect or incomplete flowers have stamen or carpels on their flowers. So this would be a situation that we think of more traditionally when we think of sexual reproduction, because you would need a male flower and a female flower, because you would need the male flower with the stamen to produce pollen and send it over to a different flower that has carpels. So that is imperfect or incomplete flowers. So the next one is monoecious plants. So monoecious plants have stamen and carpels on the same plant. So when we were up here, we were talking about these imperfect flowers. Remember, we needed two different plants. We need a male plant and a female plant, because we need one plant that has male flowers and one plant that has female flowers for the carpels. But a monoecious plant has stamen and carpels on the same plant, but not necessarily on the same flower. So an example of this would be corn. Corn has the female reproductive organs in the ear of corn, and then the stamen is the sort of the long shoot that leads up to it, the tassel. So that's monoecious plants. So then we have dioecious plants, which is stamen and carpel on separate plants. So this is similar to this imperfect or incomplete flowers. And by the way, I should differentiate these. These two up here are referring to the actual flowers, like the type of flower we're seeing, and then these two are referring to plant types. So, for example, a monoecious plant could be imperfect or perfect flowers. It could have perfect flowers or it could have imperfect flowers. Dioecious plants have stamen and carpel on separate plants, so they would have to be imperfect. So they would have to carry imperfect flowers. Because remember, imperfect flowers means that my flower has either stamen or carpel. And then dioecious plants means that I have one plant that has stamen and one plant that has carpels. But you could, for example, have a monoecious plant that has imperfect flowers. It could have male and female flowers on the same plant. So that's the sexual plant types. All right, and next up I want to talk about the three modes of reproduction. So we have three different versions. We have selfing, outcrossing, and cloning. So selfing means that it is fertilizing itself. So plant fertilizes its own egg with its own sperm. So when we're thinking about in terms of genetic diversity, what factors are we still going to get that would create genetic diversity? Because remember, we had these three things. We had crossing over, independent assortment, and random fertilization. So if I have a plant, so this plant, all of its chromosomes look the same, right? Because it's plant. So when meiosis occurs, we'll still get crossing over, right? So we're still going to get crossing over. We'll still get independent assortment. But we will not get random fertilization, because we know for a fact that this plant that is self-reproducing, self-sexually reproducing, we know that plant will be getting its gametes from itself. So there's no random fertilization. Whereas outcrossing means plant, so it would mean one plant fertilizes another. So in that situation, we have two parents instead of one parent. So we will get random fertilization. We will get crossing over. And we'll get independent assortment. So this outcrossing is going to provide the highest genetic diversity. And then the last one, and this is asexual, but just for the sake of sort of comparison, cloning means plant duplicates via mitosis, so not meiosis. This would lead to no genetic diversity. All right. So on to the next thing. So now we have the ABC model of flower development. You could also call it the whorl model. So basically the ABC model says that on a meristem, when we have an apical meristem that is going to grow into a flower, it has 4 distinct regions, and we call those whorls. WHORLS. And these whorls, I'm going to give you a top view as well. These whorls are arranged in in a concentric circle sort of format. And so you have your first whorl, second whorl, third whorl, and fourth whorl. So it works from the outside in. And this model basically says that each one of these zones, each one of these whorls, is going to be responsible for a specific part of the flower, of the sexual system. So whorl number 1 is going to become the sepals, which were these photosynthetic leaves right at the base of the flower. The second whorl is going to become the petals. The third whorl, so second from the inside, is going to become our stamen, which is going to be our male reproductive organ. And then 4, this final very middle whorl, will become our carpel, or our ovary. So this model is sort of contingent on 3 genes, and we call them gene A, gene B, and gene C. And these are homeotic genes. So this means that each one of these genes, or a combination of these genes, is going to code for something very specific. And so what we can do is we can observe the genes that are active in each of these whorls, and we found that in the first whorl, gene A is expressed. In the whorl that becomes the petals, we had gene A and gene B. And then in the third whorl that became the stamen, the male reproductive organs, we had B and C genes expressed. And then in the fourth whorl, which became the carpel or the ovaries, we just had C. So the other thing that we sort of realized from this is that A actually inhibits C, and that's why A and C are never together. So this is how the flower develops, but there is one sort of plant pathology to talk about, and that's called arabidopsis. So arabidopsis, and make sure it's in frame, it is, means that these homeotic genes will mutate and sort of they'll express in the wrong areas. So maybe you have gene B also showing up in the first whorl, which would mean if genes A and B are present, petals are going to be produced. So if in the first whorl B also gets produced, instead of producing sepals, we would produce flowers. Or petals, rather, petals. So then we would not have sepals. And so the way that arabidopsis works is it just worked out that all of these gene mutants resulted in 2 of the 4 organs not being expressed. So when you have arabidopsis, you are either expressing one of these 3 phenotypes. You just have carpels and stamens, which means you're lacking sepals and petals. Maybe you have sepals and carpels, which means you are lacking petals and stamen. Or maybe you have petals and sepals, which means you are lacking stamen and carpel. So you don't have to memorize those, but I do want you to just know the term arabidopsis. All right? All right, so this is sort of the big 2 topics from this chapter. So first we have the angiosperm life cycle, which is alteration of generations. And what does alteration of generations mean? This means that a single plant fluctuates between sporophyte, and sporophyte is 2N or diploid, and gametophyte, which is just 1N or haploid. And remember, diploid and haploid, these are chromosome numbers. So, for example, us as humans, we are diploid in the most of our body, but then our sex cells, so our sperm and eggs, are haploid because they only have 1 copy of chromosomes, but the rest of our body has 2 copies. So this is sort of a lot. I would roughly draw this in your note sheet because it is going to be pretty important. So we're going to start at the event of fertilization, and we're going to work onward. So we are going to have a pollen grain, which is the male gametophyte, which you can think of as the sperm, and it's going to combine with the female egg in the ovules. And so when the pollen grain fertilizes this egg, it's going to create the zygote. And the zygote is our first instance of diploid in the life cycle, because we had a haploid pollen grain and a haploid egg, and now we have a diploid zygote. And so that zygote is going to grow into a fruit, and that fruit will have a seed in it. And so the fruit and the seed are also going to be diploid, and eventually it will grow into the mature sporophyte. And you can think of the sporophyte as the plant as you're thinking of it. A sporophyte is like a fully developed plant. Sporophyte. So the sporophyte has 2 organs, 1 of 2 or sometimes 2 organs. So the sporophyte has the anther, which, remember, the anther was part of the stamen, and the anther is the sexual producing cells, or the sexual producing structure for males. And then we had ovaries in females that are the sexual cell producing organs in females. So the anther and the ovary are both 2N, so they're both diploid. But then, through the process of meiosis in both, meiosis is going to produce our gametophytes. We're going to have, for males, we're going to have something called a microspore, which you can think of as just one tiny little pollen granule, like the smallest pollen granule. And then in females, we're going to have the megaspore. Both of these are now haploid, so we have now entered the gametophyte stage. So remember, gametophyte equals N, and sporophyte equals 2N. So the sporophyte generation is from zygote up until this point, and the gametophyte is from this point until fertilization. So we have the male microspore and the female megaspore. So now these sexual cells are going to undergo mitosis, which means just reproduction. And so it's going to produce the pollen grain, which is just essentially a whole bunch of these microspores contained in one little package. And that is the true male gametophyte, is the pollen grain. And for females, we have the embryo sac, which is the true female gametophyte. And now that we have a pollen grain and an embryo sac, we can once again undergo fertilization of the egg to create another zygote, and it just keeps going around like that. So that's the angiosperm life cycle. I believe the last topic for this chapter, and this is a really big one, is called the double fertilization of plants. So, double fertilization means that in one reproductive instance, two fertilizations occur. And so this is something that angiosperms will do because the end goal is to fertilize the egg, but also to create a structure called the endosperm. And you see this word down here, endosperm. And the endosperm is going to help provide nutrients to the growing embryo. So let's talk about this step by step, because this is a really important topic. So first we have the male section of the plant, so the male reproductive system. So remember we had our anther, which was diploid, is going to produce microspores, which are haploid. So this was meiosis, right? And then the microspores will undergo mitosis to produce pollen grains. So a pollen grain has essentially two parts of it. It has what's called the tube cell, which is what forms the tube in stigma. And I'll go back to that picture. And then it also has a generative cell. And so the generative cell is essentially the sperm. So in one pollen grain, this generative cell will undergo one mitotic division and create two sperm cells. So we have our two sperm. And then we're also going to have a tube cell. And I'm going to quickly just go back to the first page of the notes because I want you to remember right here, we said that there's this structure called the pollen tube. And remember, we had this stigma at the end of the style. So we had this sort of shaft that goes up out of the ovary. And the pollen tube is not always there. The pollen tube is actually grown by the pollen grain. So the pollen grain is going to interact here and start growing a tube inward towards the ovary. And that is going to be the tube cell that is responsible for growing that pollen tube. So again, in this one pollen grain, we have two sperm cells and one tube cell. All right, so we now have our male reproductive cell. So now the female reproductive cell is going to get produced in the ovule. So you're going to have something called a megasporocyte in the ovule. And the megasporocyte, which is diploid, will undergo meiosis to create 4 megaspores. And remember, that was the female gametophyte. So these 4 megaspores, unfortunately, 3 of them will perish. So 3 of the megaspores are going to die, always. The one surviving, I don't know why this is, by the way, but it is. The one surviving megaspore will undergo a few mitotic divisions and create what are called haploid nuclei, which are going to be N. So we have 8 haploid nuclei now. And you don't really need to know what haploid nuclei are, just know that they're a thing and that they are 1N, they're haploid. So those nuclei are going to differentiate. So we have 8 of them as a result of 1 megaspore. So 1 of these haploid nuclei will become an egg cell. 2 of these haploid nuclei are going to become what are called synergids, and that word's right here, synergids. 3 of them, so we're up to 6 now, are going to become antipodals. And then the last 2, and this one is really important to remember, are going to become the polar nuclei. So 2 polar nuclei. All right, so now in our female reproductive sort of system, we now have an egg. We have 2 synergids. We have 3 antipodals. And then we have 2 haploid nuclei, or 2 polar nuclei. So now we have the female reproductive thing ready to go. So now we're going to get to the actual process of fertilization. So the first step is the pollen grain is going to hit the stigma. And I'll go back here again one more time. Remember, the stigma was this structure right here at the very end of the carpel. So you can think of the stigma as sort of a binding site for pollen, for the pollen grain. So the pollen is going to hit the stigma, and then this tube cell that we had in the pollen grain is going to start growing downward through the style, which was the shaft, into the down into the ovary. And this is going to be combined with the efforts of these 2 synergids. And these synergids are going to guide the tube cell and create a nice clean pollen tube right down into the ovary. So now that we're in the ovary, we have our 2 sperm cells. One of these sperm cells is going to fertilize the egg. So the sperm cell plus the egg, so that's an N plus an N, is going to create the zygote, which is now diploid, and that's going to be our embryo. But we actually have a second fertilization. So we've used one of our sperm, so now we're going to use this other sperm and these 2 polar nuclei. So we're going to take a sperm, which is N, and we're going to take 2 polar nuclei. So a polar nuclei is N, and we have 2 of them. So this is N plus N plus N, because there are 2 polar nuclei. And this structure becomes the endosperm, which is 3N or triploid. And the purpose of the endosperm, all I need you to know about it is that the endosperm provides nutrients for the embryo as it grows in the seed. So now we have a seed, and in our seed we have our embryo, and then we also will have an endosperm to provide nutrients. All right, so this was a pretty short one, but that's going to be it for chapter 38. And yeah, I will see you guys next time.